All right, I presume that we're all ready to go. This marks our first online video lecture together. This is the first time I'm doing this for this course. We'll see how this first lecture goes, and we'll try to make any necessary adaptations as we go along. But I'd like to just get right into it because we will be going over all the specifics of the class and all the technicalities and in the syllabus during our first synchronous meeting, the dates of which I've sent you on emails and will also be available on the course website on our first day of classes. So let's just go right into it. Alright, so first starting off about the genome. Before we go into the human genome, we need to go over two very distinct groups of cells in the body. They are the germ cells, okay, or collectively referred to as the germline, and then you have your somatic cells. The germline is found with the reproductive organs, all right, and is dedicated to producing a very special type of cell called a gamete. But you're probably more familiar with human gametes being referred to as the sperm and the egg. From now on, you got to be careful, or you got to be comfortable, that is, with also referring to the sperm and the egg as gametes. I know it's kind of a big word. It's a G-A-M-E-T-E-S. As you can see right along with your notes that accompany the PowerPoints. But it is what it is, and we just have to be mindful of equating this new vocabulary word with the familiar word sperm and egg. Your somatic cells are the other approximately 100 trillion. Yes, 100 trillion. I try to fathom that number. I can't. It's just such an enormous number of cells. These are the cells that make up your body. The rest of the cells that make up your body, that is, other than the very small amount of germ cells that are found within your germline, within your reproductive organs. Just remember it this way. Soma, okay, is in the word somatic, and soma equates to body. The overwhelming majority of cells in your body are somatic cells. We will begin our overview of the genome referring to somatic cells. All right? It will be specified when we begin discussing germ cells of the germline. So keep that in mind that as we go on, I will specify as to whether we're talking about gametes, namely sperm and egg. Otherwise, you just assume we're talking about good old somatic cells. All right, so finally, back to the genome. So right here, you have this representative DNA molecule, and everyone is probably familiar with this double helix, all right, shape that a double-stranded DNA molecule takes on. So here we have one strand right here. All right, and then over here we have our second strand. All right, and these strands are connected together by these lines in between. And if you think about the DNA molecule as being like a railroad track, you can think of these backbones of the strands, which I've just outlined in red, as being analogous to their tracks. And then you can think about these struts that connect them, which we will learn about their chemical composition soon enough, are analogous to the ties of a railway track. Okay? So when we're talking about the genome, we're talking about the entirety of one's DNA. All right? So if we can think about this strand here, which is in fact a double-stranded molecule, and it's going across our screen here, and the dot dot dots symbolize the fact that it's continuing on in this direction, okay, and then continuing on indefinitely in this direction as well. All right, and this is a lot of DNA. Just to put it into context for you, the amount of these little struts here, okay, or ties of the train track, if you will. There's about 6 billion of these from end to end. So obviously this diagram is abbreviated and highly cartoonish just for illustrative purposes. All right. So, but if we want to symbolize this long, double-stranded helical molecule as being all the DNA in one's body, we can then break it down into little chunks. All right, and that's what I'm going to do right now. And these little chunks, all right, which you can see one of them is marked off right here, from here to here, okay, little chunks of genome, all right, are designated as chromosomes. All right. So if one's genome is broken down into separate segments or chunks, refer to these separate segments or chunks of the entire genome as a chromosome. Okay, and this right here is just one particular chromosome. Chromosomes are extremely long and continuous stretches of your DNA that are succinct from one another, that is. So you would have another chromosome right here. And that one would keep on going, okay, until you met the point where you would have another chromosome. And here we have our other chromosome right here. All right. So let's cut out and remove this chromosome here that we're going to focus on in the middle and take a little bit closer look at that. All right, so we just excised this chromosome, okay, and within the chromosomes there are specific sequences of genes. Recall that a gene is a stretch of DNA that contains the information to build a specific protein. This protein, in turn, will play one or more roles within your body throughout your lifetime. That includes your development, okay, in utero. So these genes, even though they may specify a specific protein, those proteins can be used for lots of different things and participate in lots of different very complex biological pathways to bring about many different types of traits, all right, which may come up at different points in your life history. All right, so as a result, specific genes correlate with specific features or traits of your body. All right, so let's demarcate two genes right here. And this chromosome only has two genes, which is for us would be a gross exaggeration. We're going to talk about the breakdown of your roughly 20,000 to 25,000 genes, which are spread over your 23 pairs of chromosomes. So within a specific chromosome, you're always dealing with genes on the order of hundreds to thousands. Instead, just for illustrative purposes, I just put two. All right, you can see that the genes are not back to back. There is this gap DNA between them, and as we'll learn later on, this is also a gross distortion because the vast majority of the DNA is actually this DNA that is between the genes. All right. So right here, now we're going to say that gene number 1 is going to give way to protein number 1. All right, the mechanisms of which we will cover in a much further video. And then gene number 2 is giving way to protein number 2. And I'm sorry about the size of these letters. It's a little bit small, but this is gene number 1 and protein number 1, and here we have gene number 2 ultimately giving rise to protein number 2. All right, and then these different proteins are going to be responsible for different traits. Etc., etc., etc. All right, let's move on. Let's talk about chromosomes. So before we go any further, it needs to be mentioned that the chromosomes in the human body are located both in the nucleus and within the mitochondria, which of course are outside of the nucleus and within the cytoplasm. Okay, so here we have our little male and female right here, and we're going to blow up just one somatic cell out of their 100 trillion somatic cells. And right here you'll recognize this is our nucleus, right? Okay, and then this is a mitochondria. Okay, and the whole space here between our nuclear envelope and the plasma membrane of the cell, and the plasma membrane of the cell is collectively referred to as our cytoplasm. Okay, so the mitochondria are not in the nucleus, obviously. They're floating around out in the cytoplasm, and there's not just 3, there's a whole lot more than that. Numbers need not be specified. All right, so right here we see that in the mitochondria, within the mitochondria that is, you have the circular mitochondrial DNA, all right, or chromosomes rather, okay, and then within the nucleus we have 46 different chromosomes. I think it's worth mentioning that while you have several copies of this chromosome here in the nucleus, it is just the identical copy of the same circular mitochondrial chromosome. All right. And then over here we have our nuclear chromosomes, which as you can see have a very different shape in their most condensed form, which they only spend part of their existence in, actually a very small part of their existence in, relatively speaking. All right. All right. We will soon discuss the highly elaborate structures that chromosomes within the nucleus are organized and managed. But in contrast, let's just go over this, and you'll learn much more about this and all different types of circular genomes when you do microbiology, if you haven't already learned about it in microbiology. So in contrast, the mitochondrial chromosome and its copies are just a long strand of DNA that is connected upon itself to create a loop or lasso of DNA. These structures are a whole lot more complex that are within the nucleus. All right. And it's also worth mentioning that the vast majority of your DNA is located within the nuclear or within the nuclear envelope. For example, I just said that you have roughly 6 billion nucleotides within your genome. Only about 50,000 of them are located within this mitochondrial chromosome. All right, just to give you some comparison. All right. Okay, moving on. So here, on the topic of nuclear chromosomes, if you recall from a previous slide, the nuclear chromosomes were depicted as a group of irregularly shaped lines with stripes. We'll go back here just to show you again. Okay, here they look like little striped noodles. All right. However, before that, we had described the chromosomes in terms of long stretches of DNA, which contain many genes lined up in a specific order. All right, so even before that, we talked about chromosomes in a linear fashion, okay, with a whole bunch of genes stacked along this very linear, elongated, double-stranded DNA molecule. All right, so which of these two different comparisons are most accurate? All right, the truth of the matter is that they are both accurate. All right, so at this juncture, it's very important that you understand that the striped-tailed or striped X or noodle-like depiction of chromosomes are a highly coiled form of those long, straight strands of DNA that we originally introduced you to. Often, it is the very condensed and coiled form of the molecule that the textbooks are referring to when they use the word chromosome. Right? The highly coiled form of the chromosome only exists for the relatively small amount of time when the cells are reproducing themselves. And we're going to discuss their structures in much more detail in another video. But right here, I just wanted to show you the progression, how you go from this long, slender, double-stranded DNA molecule in a linear fashion, and as you can see right here, this specific segment of this chromosome here in its spread out elongated form represents the DNA that corresponds with a particular gene. All right. So what happens is the coils get tighter and tighter and tighter. All right. And then they start wrapping around these little octets of special proteins called histones. We'll talk about that later as well. All right. And then these little string of pearls, so to say, okay, that are a combination of the DNA molecule and these intermittent histones start to coil around themselves. Okay, so the coils start to coil, okay, and then those coils start to coil around the previously created coils, all right, okay, and then still more coiling occurs, okay, and this very, very thin structure, okay, and very, very long structure becomes increasingly shortened and increasingly stout or wide. All right, until finally, all right, they all end up being jumbled together. All right, and mind you, this is an absolutely incomprehensibly large amount of DNA being condensed, all right, within this huge structure right here, which is what we refer to as a traditional representation of the chromosome. This is the striped tails or noodle-like chromosomes that we saw before. Okay, and again, these only come out for a short amount of time, and that's when the cell is actively reproducing itself. Right? The rest of the time, the DNA is in a much more relaxed form, such as this. Okay? For simplification's sake, oftentimes in textbooks, we will depict the DNA like this. Right? And by the way, this particular chromosome right here is split up into two sister chromatids. We will give reference to that at the end of this video and then talk about it in more detail in a following video. But just as a little heads up, right here, this would be a sister chromatid. Okay, and then this one over here, okay, including both of its arms, so to say, its P arm and its Q arm, and this P arm and this Q arm, okay, are all onto themselves individual sister chromatids, all right? And it turns out that you have one big, long, linear DNA chromosome, all right, all coiled up in one of these sister chromatids, and then you have the absolutely absolutely 100% identical, big, long, linear DNA chromosome all coiled up here. And how it is that you come about creating two identical copies of a chromosome is for a future topic. So stay tuned. And then here, this is cute. This is just showing these chromosomes floating around within the nucleus of the cell, just to give you even a further level of perspective. Okay, so we went over basic chromosome structure and compared it to just plain old linear double-stranded DNA, and put chromosomes in context with the genome, and then put genes within context with chromosomes. Right? Now we're going to be talking about the actual numbers of chromosomes that you have within your nuclear envelope, and of course we call these nuclear chromosomes. Although for the most part, textbooks will forget about the fact that you have your mitochondrial chromosomes, and when they say the word chromosome, they're generally referring to these guys here. Right, your traditionally structured chromosomes. So you have 23 pairs of chromosomes equaling 46 total chromosomes. All right, 22 out of these 23 pairs of chromosomes are designated as autosomes. All right, and the reason they're designated as autosomes is that autosomes are shared by both males and females. They do not possess any genes that command sexual differentiation. Okay. Now you might be wondering now why I mentioned genes commanding something, and some of you might have said, well, Matt, you just told me that genes are responsible for making a certain protein product. But keep in mind that eventually that protein product is going to go and do something in the cell. All right? It will give way to a certain trait or participate in creating some traits or facilitate some type of chemical reaction, et cetera, et cetera, et cetera. Okay? And it will also be involved extensively in controlling the differentiation of certainly some cells within the body. And in this case, the autosomes, these 22 genes that are 22 chromosomes rather, 22 chromosome types that you have in common between males and females, are not involved with bringing about any of these differentiations that are involved in sex and reproduction, right? So again, I can't say this enough, and I would suggest if this is somewhat new to you, or you learned it at one time and you've forgotten it, and maybe you learned it again and forgotten it, and learned it again and forgotten it, just copy this over as many times as you can bear. All right. There's nothing wrong with rote memorization to start off with. Actually, that's probably the preferred way to at least begin studying. So we already said that of the 23 pairs of chromosomes equaling 46 different total chromosomes, you have 22 pairs of autosomes, and then of course 23 minus 22 is 1, so you have 1 pair of sex chromosomes left. The genes in the sex chromosomes do specify commands for sexual differentiation. All right, and when I say sexual differentiation, I mean creation of reproductive organs and other traits and features of our bodies that differentiate males from females. All right, females have 2 X chromosomes, whereas males have 1 X chromosome and 1 Y chromosome. There are exceptions to that, and we will certainly be learning about them later on in class. Now this is an extremely important concept, and I want you to keep on going back over and over and over again until you really get this. And if you're still having problems getting this concept, then please don't hesitate to contact me as soon as possible. We can meet on campus, we can talk over line or over the internet using a synchronous connection with a roommate. We can correspond back and forth through email, so don't hesitate to reach out to me on that, because this really is important that you get this concept. There are 23 pairs of chromosomes, making a total of 46 chromosomes. But I would like to add one more vocabulary word into that equation, and that is homologous chromosomes. So now you can say there are 23 pairs of homologous chromosomes, equaling a total of 46 total chromosomes. Right? Homologous chromosomes have the same types of genes in the same order. They have the same types of genes. Uh-oh, we got a little gap right there. All right, so thank God for so many different versions of PowerPoint. So homologous chromosomes have the same types of genes in the same order. So remember we gave that diagram of that DNA molecule? Okay, supposed to be representing a very crude chromosome. Here's its double-stranded DNA molecule. All right, and we would have gene number 1, okay, and right here we would have gene number 2, okay, and then right here we would have gene number 3. I think you guys know where this is going, right? And then right here we have gene number 4. Gene number 1 is gene number 1. That's its identity. Gene number 2 is gene number 2. Gene number 3 is gene number 3. And gene number 4 is gene number 4. Okay? They give rise to a specific protein. In this case, gene number 4 gives rise to protein number 4. Gene number 3 gives rise to protein number 3. Gene number 2 gives rise to protein number 2, and gene number 1 gives rise to protein number 1. All right, so that's what we mean by the same types of genes in the same order, okay, along the chromosome. All right, so however, homologous chromosomes are not identical. They are not the same. And they're not the same because these gene number 1s from one chromosome in a pair to the other homologous chromosome within a pair can be slightly different from one another. And these gene number 2s from one homologous chromosome in a pair to another homologous chromosome within that pair can be slightly different from one another. And gene number 3 and one homologous chromosome in a pair can be slightly different from another homologous chromosome in a pair, etc., etc., etc. All right? Okay. Homologous chromosomes contain the same types of genes, but the genes can be slightly different from one another. Now, what I want you to do is that phrase right there, homologous chromosomes have the same types of genes in the same order. I want you to say that 3 times out loud, over and over again, while you're watching the screen. And then I want you to turn away from your computer, and I want you to say it 3 or 4 times more out loud. All right? And then I want you to think about it, and go back and of course do the readings. All right? And if it's still an issue, then please come in and see me. But this is something that I really want you to dig deep ruts in your mind with regard to, because it's really going to help you later on deal with a lot of rather not complex, but sometimes frustrating lines of logic. All right, if you can just get this basic concept down of homologous chromosomes. We'll go over some pictures soon enough to help you visualize it. So let's talk about another very important concept, and that is of alleles. Homologous chromosomes have the same types of genes in the same order. I've said that over and over and over again. It's important. But we also said the genes can be slightly different from one another. All right? These slight differences among the same type of gene, okay, and which, by the way, are located in the same order and are also at the same locations within the different homologous chromosomes. We call these locations loci. The plural locus would be the singular. This just means the relative place within the chromosome that a specific gene exists. Okay, well, the genes of the same type in homologous chromosomes will be at the same location because, of course, they are in the same sequence to one. Okay, but these slight differences of these same types of genes at the same locations are called alleles. Okay. And what makes these genes slightly different from one another, we will get into in the second lecture when we talk about the specific structure of it. Okay, but right now you just have to know that these slight differences among the same type of gene, meaning, for example, gene number 1, gene number 2, gene number 3, gene number 4, that make proteins number 1, number 2, number 3, number 4, respectively. All right? But they have slightly different aspects to them, okay, in their basic design. All right? So when you're talking about alleles, instead of talking about protein number or gene number 1, gene number 2, gene number 3, gene number 4, you might want to think about them in terms of gene number 1A versus its allele gene number 1B, or another allele gene number 1C. Or think about gene number 2 in the same terms. It has alleles gene number 2A, gene number 2B, or gene number 2C. Gene numbers 2A, 2B, and 2C are all the same type of gene because they're all number 2 genes, but they all have different varieties of the A, B, and C type. Right? I know it's confusing with all these words, but we just need to put the words out there so you can go back and use these slides to study from. So let's go ahead and look at some pictures. Alright, so what you're looking at here is a karyotype of a human being. Basically, it's a photomicrograph. And you might be asking, what's a photomicrograph? Well, it's just a photograph that you take under a microscope. And it's a photomicrograph of all 22 pairs of homologous autosomes, and then, of course, your pair of sex chromosomes of this particular person's genome. This particular karyotype is a far more fancy one than most, because the scientist who made it has added fluorescent dyes that bind to very specific areas on the chromosome. These specific areas correspond with large groups of genes, by the way. The specific or special technique of this kind of karyotyping is known as FISH, F-I-S-H. It's an acronym that stands for fluorescent in situ hybridization. Now, you don't need to know that, but we will be going back to that at a later time, so I just want to throw it out there for now. But anyways, we'll discuss the specifics of how karyotypes are made and how they are used in clinical medicine at a later date. But right now I want you to notice that the homologous chromosomes in each pair are the same length, except for one pair, okay? And you can probably already see which one it is. It's the sex chromosomes, X and Y. Alright, so this individual here is obviously a male. Alright. But the reality is that the sex chromosomes in X and Y particularly are not technically homologous because they have different lengths and they have many different sets of genes. Okay, they're quite different. But they are paired together because you always pair together the sex chromosomes. But all of these other guys are homologous. Same types of genes in the same orders, but little differences maybe in those particular genes from one chromosome to another. We call those little variations or differences in genes of the same type alleles. Right? So I'll also take a look at the colors here. Okay, so notice how the colored bands match up between the two homologous chromosomes of each pair. Okay, each pair of colored bands, for example, this magenta band right here corresponds with this magenta band. This magenta band corresponds with this magenta band. Alright, this little blue band right here corresponds with this blue band right here. This red band here corresponds with this red band here. That's what I mean, okay, by that. Okay, they represent the same areas of the chromosome with the same sets of many genes. And within these little areas, you're dealing with, at least 50 genes backed up, or lined up rather, back to back. Okay, just in these tiny little bands here. Okay, so you're not actually looking at individual genes lighting up. We would be pretty simple organisms if that was the fact. But within each of these separate areas, you'll probably see 50 or more separate genes back to back, give or take. The genes within the adjacent colored bands on homologous chromosomes are the same types of genes, and they are in the same order. Alright, so let's say within this little banding pattern here, there are 50 genes back to back in each one. Those 50 genes are the same type of genes in the same order. Okay, just like all of the rest of the genes along both of these homologous chromosomes, this in particular is number 8, are all the same types of genes and they're all in the same way. The position of a single gene on either chromosome is called its locus. The plural is loci. The locus of the same gene on the homologous chromosome will be exactly the same. Okay, so within this area, if you have gene number A right here, okay, gene number A will also be found in the same area on this homologous chromosome. Now, gene number A may be of a different variety on this component or segment area of the chromosome than it is over here, but they will be the generally same type of gene giving rise to the same type of protein, all right, and being in the same location. That's what we mean by homologous. Basically, essentially the same, but not quite perfectly exactly the same. And we'll talk about, when we talk about the actual structure of the DNA molecule, we'll get into more specifics on how alleles differ from each other in small ways. Okay. Another aspect that I want you to appreciate of this specific karyotype is the fact that they are numbered, okay, in order from 1 to 22, so that number 1 is the longest set of homologous chromosomes, and number 22 is the shortest, and everything else is proportionally in between. All right, so that's how they number chromosomes. It's according to the total length of the chromosomes, where number 1 is the longest and number 22 is the shortest, and everyone else in between is a function of their relative length. So keep that in mind when people give a shout out to the number of the chromosomes in your mind. You can kind of get a sense of how big the thing is, and then also how many different genes it contains as well. So now let's look at another karyotype right here. This is a much more this is a much more basic karyotype. It's not one that was made with fluorescent in situ hybridization or fish. It was one that was made with a type of stain called giesma, which they mention in the book, and it just creates this monotone or monochromatic rather banding pattern, sometimes called G banding in the laboratory. Let's focus in on just one of these chromosomes. We'll take a look at chromosome number 3. Okay. So if we were to blow up these two homologous chromosomes, both of the number 3 type, okay, we'll see two large chromosomes side by side. Okay. And the different color stripes, here we have purple, here we have green, here we have kind of a cyan color, represent different specific genes. All right, but also keep in mind that no single gene would ever take up this much space on a real chromosome. Okay, these genes are made extra large just for the purpose of demonstration. Okay, an actual gene in the context of an entire chromosome would be much too small to actually even really put a dot on it. You can really only express it in a very figurative way, which we've done here. Notice that the bands at the same locations are slightly different between the two homologous chromosomes. Okay. And their difference is in how they're shaded, because right here, even though this purple banding is in the same location on this chromosome as it is on this chromosome, they have a different overall shade to them. So we have light purple here, okay, and we have dark purple here. Okay. The reason that they did this is because while they are the same type of gene in that they are both purple genes, okay, they have slight differences. So this dark purple gene versus the light purple gene are in fact alleles of one another. Okay? And that's what they mean right here. So homologous regions code for the same gene. Okay? And they would be in fact alleles of one another. Now here's another thing I want you to take a look at. Another aspect of this diagram I want you to notice is that the shape of the individual blown-up chromosomes, they aren't just like lines or noodles as they are in the karyotype. See right here where each one just kind of looks like a line or a noodle. This one looks kind of like a noodle. Okay. In fact, instead they have this big X shape to them. Okay. And this is important because these chromosomes actually possess two exact replicas of their own individual DNA. Remember when I mentioned before the previous diagram, okay, is what you actually have here is two identical sister chromatids on this homologous chromosome, and then on the other homologous chromosome, all right, you have two other exact replicas of one another. Okay, two sister chromatids attached right here. Okay, and they are both attached right at the hip, so to say, okay, at their central region, which is called the centromere. Okay. So usually you cannot visualize these individual sister chromatids on these karyotypes. Here you can almost make it out, okay, that there are two X-like shapes there, but sometimes it's very difficult to tell. So when you're looking at these karyotypes, it's important to realize that within each chromosome, within each homologous chromosome, there are two completely 100% identical sister chromatids. Okay, and so in reality, it has an X-like shape, even though you may not be able to directly appreciate it. We're going to get more detail about the architecture of chromosomes soon enough. However, before we do that, we need to take a closer look at the structure and replication of the DNA molecule itself, and that is what the Part 2 sequence for Chapter 2 of the Human Genome and Chromosomal Basis of Heredity out of Thompson and Thompson Genetics and Medicine will be focused on. So I appreciate your attention, and I will be meeting with you all soon enough. And I will talk to you soon. Bye-bye.